Okay, and welcome to the next section of the course. This one is on, as they put on the syllabus, GMOs, which is kind of the term that we use because people understand what it means. It's a term I don't like to use because for me, I think of this a little bit differently. We'll talk about that as we go through the next 4 lectures. So my name's Kevin Folta. I'm a professor over in horticultural sciences and also the chair of the department. And the way that you can find me is right up here on the screen. You can usually email is best. And we'll talk about some of the other aspects of the things I do as we move along through the semester. But the main point of this section is to familiarize you with the information that's currently available in the area of genetic improvement of crop plants and animals. Just what are genetically modified organisms? How do we do it? What is genetic engineering? And what are the strengths and limitations? What are the risks and benefits as told to us by the peer-reviewed literature? And that's really what I'm interested in. Over the last 12 years, now 16 years, I've spent a lot of time studying this topic and a lot of time speaking with the public on this topic. I actually started studying it in 1977. I was 10 years old and I took a book out of the library on recombinant DNA technology. And back then it was a very thin book, but it was interesting from a kid's perspective that I didn't understand what was happening, but I knew it would be important. And very fortunate that so many years later can still work in this space. But just for what it's worth, my laboratory does a couple of different activities. We study how we can change the way plants grow, the way the nutrients they contain, and the flavors and colors by using different wave bands of light. We also study the genes associated with aromas in small fruits. So we study the genes that are associated with flavors in things like strawberries. Our goal is to be able to enhance breeding and the speed of breeding by getting all the genes in one place to make better improved crops. So there's no part of my research program that is directly related to release of commercial GM crops. We don't do that in my lab. But still, it's a topic I study and understand quite well. And just for full transparency, all my funding and reimbursements are on there because it's always important to put that information out there when discussing this topic. So over the next 4 weeks, we'll take a look at 4 different topics. Well, first today we'll study crop genetic improvement. Why does it matter? Why is it important that we are able to improve plants for human consumption and other human uses? We'll talk about, next week, we'll talk about the main traits that are used and the traits that we'll be seeing in the future. The plants that we'll see coming up to solve problems for people and the planet. In the third week, we'll have some fun. We'll talk about the public controversy. We'll do a lot of myth busting. We'll talk about what are the aspects of this technology that people have found objectionable. What's reality and what's just myth? And we'll talk about that in week 3. Week 4, I was hoping to put together group projects and presentations, possibly take on a short exam. Exactly how we'll do that is yet to be seen. But what I imagine is I'd like to be able to have people in this class be able to come forward with ideas as to what is a solution they would like to solve with regards to food, farming, the environment, and how could we use this technology as an optimal way to meet that challenge. And I think that would be a pretty easy way to do it. So I always like to start out this discussion by asking an important values question. What's important to you about food and farming? I don't know, there's not many of us actually in the room, but what do you think? What's important to you?
It's one of the main necessities that humanity needs. It's always going to need it, so it's better to take care of it.
So taking care of
The quality of food.
Okay, the quality. So the one answer was taking care of the quality of food, ensuring the quality of food. What else do we think of?
Well, like she said, food is something that we're always going to need, and it's something that's, once we really figured out how to farm, 10,000 years ago, it's something that's constantly evolved. But I feel like now, especially with our population growing so much, we're going to have to find a way to work in balance of the environment, keeping everything, still livable, still a livable habitat, I guess, for Earth, so we got to live here and be able to produce a large amount of food at the same time.
So you bring up a couple really important points. We talk about the idea of more people and being able to feed people, but then also the environment, our environmental impact of farming. And if I have a general class, and I've asked this question to thousands of different groups of people. And in a general audience like this normally would be, people would say, well, I worry about the quality of food. I'd like to make sure that it's good quality, but also safe. I'd like to make sure there's lots of it and that I can afford it. And so it boils down to price, quality, amount, and the other thing, that I like it. And so those are the qualities that we usually hear discussed inside a typical group scenario. But we don't talk about the other aspects of food and farming that are really critical for us to maintain. And you hit on one with environment. And let me tell you what keeps me up at night as a scientist. And the reason that I do this and the reason I discuss a contentious topic like genetic engineering with public audiences. It's really important. It's because things I worry about is I worry about care of animals, how they're treated humanely and how they're farmed. I worry about conservation in the environment. I do worry about the impacts of farming. But at the same time, I worry about how do we keep farmers in business? How do we take the 1% of the population that feeds us, who have an average age of 58 years old, and how do we ensure that they have sufficient means to stay in business and actually make it a lucrative business where more people are attracted to the profession? I worry about food safety and worry about food for consumers here in the U.S., that people have more choices and better choices. It's clear that people frequently have too many calories and that they're eating the wrong foods, and that we could be helping our diets and our longevity by making better choices, which could include more fruits and vegetables. And of course, one other thing is how do we extend the bounty that we have in the industrialized world to those who have nothing? The fact that 21,000 people will starve today is really a sad statement. And do we have technologies and opportunities to take care of them too? So, whereas when I normally ask a public audience, what's important to you, it tends to be very about us, about our families, about us. And really what we have to start to do is think about this on a much bigger level. What are the big issues? What are the long-term issues? What are the issues that leave the borders of this country? And when we start to frame this question this way, it means that we definitely have a place open for crop genetic improvement. It's one of the areas we need to. Yes.
Does that answer kind of like I took a humanities class, and I mean we're talking about how like in the United States we're very like secluded. We just think about kind of like us, ourselves, and then like just like our like immediate family. But then like, and especially like in Latin America, like it's a lot more like wider. Like you don't just see it yourself. It's more like your whole like extended family. So do you know if that answer kind of changes depending on like the?
Yeah. So the question was, is this really kind of insular view of everything, something that's specific to American culture, and that you see other cultures that tend to look a little wider into extended families and perhaps environment? And I think it's hard to say, and I think it changes with time. And I do think it's getting better. I think that talking to people in college these days and younger, there is more of an interest in environment, where their food comes from, that we didn't have when I was a kid, when I was coming up through college. So I do think that things are getting better. I do think that it is important, and I know that for most scientists, we got into this business and stayed in universities rather than companies because we were worried about the planet we live on and the people that are stuck to it. And are there ways that we can make that experience a little bit better by doing what we do? And that's why we got really excited to be able to remedy some of these situations. And I always like to show this slide up front because I want everyone in an audience to understand where I'm coming from. I'm talking about genetic engineering, but I'm thinking about these as my endpoints. It's not about the quarterly earnings or any company, which is what people tend to conflate with this technology. This is definitely some very important objectives. And normally I ask an audience what crops are genetically engineered. And maybe you can just think through in your head which ones we know currently are genetically engineered. There's really just several. There's not that many. Most people, if I ask a public audience, they'll say, well, let's see, bananas and strawberries and watermelons. No, nothing like that at all. It's really, it's corn. What else?
Soybeans.
Soybeans. Corn and soybeans. No, not wheat. That comes up all the time too.
What about papaya? Papaya
is genetically engineered, right? And we'll talk about that one next week extensively. What else? So we said corn, soy, papaya. And we got alfalfa, is one might not get right away, sugar beets, cotton, I'm sorry. Well, potatoes are genetically engineered, not available very much on the market yet, but coming soon. We'll talk about that one next week too. So now we have, and I lost my train of thought on which canola is another one. And it seems like there's one more, squash. And now there's a genetically engineered apple that will be out too. So it's just a very limited number of crops. And what we get from those, only 4 that we usually eat. And the thing that's important to remember, really just products from 4 of those plants. It's corn, soy, sugar beets, and canola. So it's typically the oils we get from canola, the dusting of sugar that we get from sugar beets, the little bit of cornstarch that's used inside processed foods, soy oil, soybean solids, soy milk, that kind of thing. So it's a number of 4 different crops that are in our diet that have been improved with genetic engineering. Very small number. But let's go ahead going forward. And of course, papaya and squash, a very small amount of that. So I usually don't include that in that discussion. But what are some, and we could dwell on objections, but a small group like this, let's just move ahead. Why do we have to genetically improve plants? And this is a good question. I can ask you too. What do you think? Why do we have to genetically improve plants?
Because of all the bacteria and all the fungi and all the things that are constantly changing, creating resistance to so many other things that like pesticides and stuff like that. They're just creating resistance to it.
Okay, so one answer is an evolving resistance to pathogens. And that's a really, pests and pathogens, right? That's a really good answer. What else? What else we got?
Like you said earlier, we have such a small population of farmers that, I mean, if you genetically improve a plant so where you don't have to like constantly be overlooking it, then it makes it easier for the farmer to like be able to rely on like the genes of the plant to do something instead of him having.
Okay, so something that would aid a farmer with production, maybe having fewer inputs, maybe less insecticide or different herbicides, maybe not having to till, maybe not having to use as much water, or maybe getting a little better yield. So there's all kinds of good production traits that we can think of as well. But if you think about, maybe the next slide makes this a little more clear. Here's some more things we can think about. Just plant genetic improvement. And we're not talking about genetic engineering, just genetic improvement gives us more different varieties, helps us get better yields, growth under extreme conditions, improve nutrition. And that means better vitamins and nutrient content, as well as the removal of anti-nutrients, things that actually can be harmful. And then safer products, such as suppression of allergies, that kind of thing. So the reason we have to do plant genetic improvement is to make better varieties, things that work better for farmers, things that are safer for us, and maybe things that fit better into a production scenario. The problem is that when today's culture, we think plant genetic improvement is something very different. Actually something kind of nefarious, something that we shouldn't be doing. And so this is where we have a real problem with something that really is an important part of the human experience, and that's plant genetic improvement, and what people think it is. And so before we can have a really important discussion about genetic engineering and how it works, we have to understand the way that plants have been genetically improved for thousands of years. Really the question is, what is natural? And this always comes up when I ask what are the objections. People say, well, genetic engineering, it's not natural. I don't want it in my food. I don't believe in it. It's not natural. But what I'd like to show you in the next few slides are ways that humans intervened in changing the genetics of plants that totally are not natural. First thing that's happened is that humans have taken plants from where they belonged, where they evolved, and moved them to places where they don't belong in order to cultivate them. And this is really a surprising map. It's a map that shows the center of origin of all the major crop plants, at least most of the things that we consume on a regular basis. And what you see is that the majority of crops that we come in contact with didn't come from North America. There's sunflowers, blueberries, there's a few other things that do show up in North America, but in general, pretty thin. And I always like to show this slide in Canada. What do you got? And they say hockey, and we put gravy on French fries. Yeah, you got me there. But the basic idea is that everything that we consume is a foreigner on our soil. and things that we have growing on our farms and in our grocery store really came from another place on the planet. And that's important for us to remember when we have discussions about the way that farming affects ecosystems, the way that farming affects pollinators. These plants don't belong here naturally. They were moved here because humans wanted to eat and wanted a diversity of important fruits and vegetables in their diet. But it's not just where they came from that changed. Farming and variety improvement has also changed plants radically from what they were as wild weeds or wild trees or shrubs to what we now see as highly cultivated, very valuable crop species. This slide is really important because it shows three good examples of how humans have changed plant varieties. In the upper left-hand corner, you see what's called teosinte. Teosinte is a bushy plant that looks like corn that's in the kind of the heel of Mexico, and that gray circle there is a quarter that's next to what is a very small ear of what are a couple kernels and casings that are hard as rock. In over 16,000 years, maybe a little longer, humans changed that teosinte plant into modern corn just by breeding and selection. And it wasn't even breeding that was done on purpose. You can imagine human beings maybe 16,000 years ago were out collecting teosinte, and in bringing it home would drop some seeds and see plants grow and realize that they were in control of where those plants might be. They may decide to plant them close to home. And then one day came outside and all the plants were gone except for one, the one that survived the insect attack. They just made a genetic modification. They selected the plant, or nature selected the plant, that now gave them a trait that they thought would be very valuable, surviving insects. Years later, maybe one had 2 rows of kernels instead of 1, and any farmer would select that one to feed their family in subsequent years. It was this glacial process of breeding and selection, of identifying the best ones in subsequent generations, that genetically modified teosinte into modern corn. It was all human facilitated. We saw the same thing happen with tomatoes. Look at small little bulbs on the ground that grow on vines that taste horrible. These orange ones here from the Galapagos, the major chemical is the same chemical as in cat urine. But breeding and selection gave us size, took out those nasty chemicals, and gave us tremendous variety in different tomato varieties that we know today. The natural banana, as it grows in the understories of places throughout Indonesia, is very small and loaded with seeds, has very little banana pulp. Yet the bananas that we enjoy today are large and have actually an entire extra set of chromosomes. Humans made these different selections and these different genetic improvements. And so whenever we start some sort of discussion of genetic engineering, it's important for us to keep an idea of how humans have changed plants, both from where they were and also their quality. But it's not just plants. This slide shows the way that we've changed chickens over the last 50 years. And if you look at the top row, these are broilers from 1957 at 43 days to 85 days after hatching, compared to 2001 from 43 days to 85 days after hatching. And you can see the tremendous difference in the way that the muscle is changed on these different carcasses. And what it tells us is that the difference that we're seeing is because of better genetics and probably some better nutrition and feeding habits. But the basic idea remains the same. We've been changing genetics of plants and animals. One of the greatest innovations was probably artificial insemination that just made it possible to produce much higher quality milk from higher quality cattle. This example of this particular bull here, Round Oak Rag Apple Elevation, born in 1965, had 6.5 million great-granddaughters because he was such a good producer of high-quality offspring. So not natural, again, human intervention into the process of developing high-quality food. So the reason I wanted to show those last slides is to remind you that everything that we eat is not natural, that everything comes from someplace else and has been radically genetically modified by humans, changed in ways that mixed genetics, and did so in ways that we didn't even understand what we were doing, yet we thrived as a species, as a human race, because of genetic changes in food. That it was that early agriculture that allowed us to move from hunters and gatherers to be able to partition to people to be farmers, specialize as farmers, others to take on other jobs that would allow us to grow civilizations. So what are the techniques that we use in plant genetic improvement? And what I'd like to do now over the next maybe 10 minutes is familiarize you with different techniques that we use to make plants better. And this isn't genetic engineering. What I really would like to do is set up a contrast for how this works versus how genetic engineering techniques work. The first slide I need to show you reminds you perhaps of something you maybe haven't seen since a biology class, but that's the central dogma of molecular biology. For some of you, it's familiar. This is the process where we think about DNA as the hard blueprint that resides in the nucleus of a cell. The DNA contains that important code that stays there located in the nucleus. Through a process called transcription, its information is copied onto an intermediate called RNA. That RNA contains the same information as DNA but leaves the nucleus and moves into the rest of the cell, where it moves out onto ribosomes. Some of you may remember that for the process of translation, where that information encoded in the RNA that originated from DNA now is translated into a protein. The proteins of the cell are the catalytic and structural elements. They do the work. They run the reactions. They form the structures. So the traits all come from, in most cases anyway, from the existence of proteins. And then the way to think about this is to put this into a computational context. DNA is like your hard drive. It's stuck inside the machine. You never see it if you're lucky. It's locked away inside the case, protected. The information on that hard drive can be copied onto a transient form, the USB drive, which that's it's fragile. I find one in my washer every week. It's fragile. It's transient. The information on it usually isn't meant to stick around or be permanent. That information can then be taken somewhere else to do work, such as taking a file to work because you want to print it out in color. So this is a way in which it really simulates the central dogma of molecular biology. Hard information to a transitory intermediate that can be taken somewhere else to do some kind of job. And when we're talking about genetic engineering especially, and we'll talk about this next week, we have ways of placing material on the hard drive, eliminating the USB drive, and then affecting what happens downstream at the printer. We'll talk about that later. The big trick, though, is for us to now focus on what are the other ways that we've traditionally genetically engineered crops. What are ways that we've added or changed the information on the plant's hard drive to give us a different trait that came out the other side? And the next slides will show that. One of the basic ways was traditional breeding through plant sex. How do you take two plants with different characteristics and homogenize them in one genetic background? It's pretty simple to do. You can use a variety of techniques to remove pollen from the male plant and place it on the pistillate parts of the female plants and make those genetic crosses, kind of mixing genes from two different plants with very different characteristics. This is important in plant breeding because in the process of plant breeding, we're frequently trying to combine favorable genetic traits from multiple different plants into one single genetic background. Now, this is done by plant breeders, but certainly happens in nature, that you see plants combining across wide varieties or wide genetic differences as DNA is carried on pollen in the wind or maybe by pollinators. But this is the basic idea. This is the first way in which we can improve plants genetically. It's just with cross-pollination and selection. Humans started to play with this idea, and back in the 1800s noticed that you could start to get what's called inbreeding depression, that if you crossed a plant, or many different plants, if you crossed a plant against itself, every subsequent generation had a little less vigor. Later on, we would see why this would happen, because genetically, you're getting more and more homozygous, as we would say in genetics class, that we're getting more and more of the same genes because we're just crossing with ourselves. This frequently happens inside plants. And it's an important way for us to develop new varieties because we take this plant that's genetically complex and we make it more and more simple. You almost can think about this as dogs, that the more you inbreed them, the less vigor they have, the more susceptible they are to disease and strange disorders. The mutts have the hybrid vigor, right? But that's what we see in plants as well. And here's a good example. Here are two different inbreds, B73 and Mo17 corn, Missouri 17, that these two together have very low vigor, yet when crossed together make this beautiful hybrid. And that's because you're creating these plants that have inbreeding depression from very narrow genetics in two very different backgrounds that when you combine them, restore and augment the genetic variation that causes this vigorous intermediate. So this is one way in which humans were intermingling in plant genetic manipulation, that you were creating these very strange inbreds and then combining them together to get very strong outcomes. This is what drove the initiation of the seed industry. If you look at corn yields from the 1800s, you're getting maybe 15 to 25 bushels per acre if you were lucky. Around about the early 1920s, this introduction of hybrids, being able to combine two different corn types that together gave a very favorable intermediate, really increased production. And as time would go on, the production of better inbreds and improved hybrids really led to an acceleration of the ability to produce higher yields in corn. Biotechnology has just a little bit of an augmentation on the end of this and really doesn't change yield very much. We'll talk about that next week. But other ways to genetically improve plants. Making nonspecific crosses. We can cross two different species together and get an outcome. And the problem that you have with this is that when you're crossing very different genetics is that you might do this because you have a plant that has very good resistance to disease and another one that has very good quality after you harvest it. And you're trying to get all those favorable traits in one background. The problem is that's difficult to do. It's hard to move just one single trait without bringing along all the deleterious traits around it. Here's a good example that we can think about from what is it? Walnut trees, or no, American chestnut. So American chestnut used to dominate the Appalachian Mountains. That a squirrel who wanted to walk from Georgia to Maine could do it across the top of American chestnut. But in the late 1800s, a disease was brought in and introduced, that fungal disease that destroyed all of the American chestnuts. Chinese chestnuts don't get that disease. So one thought would be you could cross an American chestnut with a Chinese chestnut and then get a resistant offspring, right? The problem is that offspring is half Chinese chestnut, half American chestnut. But what you can do then is you can cross that half and half back against the American chestnut and get something that then would be 3/4 American chestnut. Then you could cross that one back against American chestnut and get something that's 7/8. And you can see after several generations, you can get something that almost would be completely American chestnut, maybe keeping that Chinese chestnut resistance trait in that one little bit of the original piece that's in there. But you can see the problem. Breeding chestnut trees is a slow process. You might get one generation every decade or two, right? So traditional breeding can be a very slow and laborious process, and that nonspecific crosses is a way to move a single gene or a single important trait. It can be very slow. We can speed this process with DNA tricks. And what we're learning now is ways that we can use what's called molecular-assisted breeding or marker-assisted breeding in the process of plant genetic improvement. The next slide I'll show you comes from my laboratory and shows you some ways in which we can move a trait associated with the peach flavor in strawberries. So what this is showing you, this is called an agarose gel. It's a matrix that we can place DNA at one end of it and move it in an electrical field and sort it by size. So on the edges you see two different sets of white bands on a black background. And each one of those represents a different piece of DNA going from small on the bottom to large on the top. In between, you're looking at a series of products that were generated through amplification of a very specific DNA piece using the methods that we use in, say, forensics when we're trying to match a criminal to a crime. This is what is called PCR, for polymerase chain reaction. We performed PCR around a gene that we thought would have a role in production of the peach flavors in strawberries. Strawberries have some peach notes, and consumers find them very favorable. On the far left, you see Mara and Eliana. Mara is a parent that does not produce the peach flavor. Eliana does. And so what marker-assisted breeding allows us to do is to cross Mara with Eliana, and then in the next generation, which is shown here as the next plant 5 through plant 19, in the next generation, we can predict which plants will carry the ability to produce that favorable peach note. That means we can get rid of the rest of them. And that means we can eliminate those from the breeding program. So this is one way that we can accelerate breeding efforts, because we can add a gene. We know the sequence of the gene, and therefore we can track it through generations. And that allows us to breed plants faster.
So then from there, would you grab one of the plants that has like a peach flavor and then breed it again with the Eliana?
We could do that. So the question was, can you take one of them with a peach flavor and breed it back against Eliana? And that might be a really good idea. The problem is that you're losing a little bit of the genetic variability that's there. And so maybe you would want to find a different parent that would have the peach flavor, like on this slide. Maybe you could take Sweet Charlie, which has peach flavor and also other important traits. So it allows us to better predict parents, and parents that will carry favorable traits without having to grow that plant and test it. So this is a really nice trick for us to do to accelerate plant breeding. The other thing that we see is genetic vigor that comes from wide genetic crosses. And this is a good example, that strawberries that we eat today are a combination of a North American strawberry that was very slight, very small fruit that didn't taste good, and a Chilean fruit that was a little bit bigger. And a spy and colonists brought these back to France, where they got together in a botanical garden in Versailles and gave rise to the modern strawberry. Again, this could never have happened naturally. It happened because humans intervened and muddled in plant genetics. The other thing that we can do is make crosses that are impossible. So if you cross two things together that would never be able to give you a fertile offspring, sometimes we can take the resulting embryo that normally wouldn't be viable because of its highly disparate genetics of the parent, and place it into a hospitable environment of a culture dish, and be able to provide it with nutrients and sucrose and everything it needs to be happy. And so once in a while, those embryos will regenerate into an entire plant that then is viable. So this is, again, something that couldn't happen in nature, but is an important part of plant breeding and in plant genetic improvement. The other thing we can do is create polyploids. Polyploids are, again, plants with too many chromosomes. So in humans, we tend to have the same number of chromosomes, a set we got from mom, a set we got from dad. Plants, well, they can live with many sets, and in some cases, they'll inherit several from mom and several from dad. Strawberries get 4 from mom and 4 from dad. So we can induce polyploidization by using specific kinds of chemicals. And it's another way that plant breeders have been able to improve plants. Now, polyploids tend to be bigger. They tend to have larger fruit, larger flowers, and tend to be more vigorous. Another good example is of how we get seedlessness, because once you have a polyploid, it's easy to get seedlessness in the subsequent generation. In watermelons, for instance, if you double the chromosomes in one generation, in one background, and then breed it against a diploid, where one that has one set of chromosomes from mom, one from dad, and you breed that against one that's got 2 from mom and 2 from dad, it creates one that has 3 sets of chromosomes, and the offspring are sterile. So when you look at bananas, watermelons, these are all examples of triploids that the seeds are there, the embryos are there, they just didn't develop, just weren't viable. Yes?
Question? Isn't it risky that you're creating sterile offspring?
I'm sorry, isn't there a...
It's risky that you're creating sterile offspring.
So isn't it risky to create sterile offspring? Well, in this case, but these are for human consumption, and humans like seedlessness. And so in this case, you're getting a horticultural trait that is a consumer trait that the consumer wants. At the same time, the genetics of having the 2 parents are completely independent.
So you're still creating both parents, but for the consumer, you do the sterile.
Right. Yes. So we actually breed diploid and tetraploid watermelons independently. That then you test to see how they combine, and do they make a nice product that's seedless. So yeah, it's strictly a human-induced block, just by strategic crosses. Other things we do are radiation or mutation breeding, where we can use radiation or chemicals to bombard DNA with insults that break DNA in funny places, cause chromosomal rearrangements, large-scale deletions. This has been done since the 1940s and 50s and is a significant player in a number of key plant traits, such as some of the high lycopene or dark red and ruby red grapefruits. There's a number of traits in barley and other grains that came from mutation breeding. Quite a few plants have been altered by this kind of random treatment of plants with radiation or chemicals. So that's another really interesting one. The other thing we can do is regenerate plants from single cells. So plants in culture, this is a piece of a plant that has been placed into medium to cause it to create what's called callus. Callus is just a blob of dedifferentiated cells. And from that can come new shoots. And we'll talk about this more in a minute. But sometimes those new shoots have genetic changes that occur because of going through this process. And they produce what's called somoclonal variation, that they're changes in plants that are brought on because of the culture conditions and regenerating through this intermediate culture step. Again, another way to create genetic variation. Let me scoot ahead here to just sum up this part of the lecture because I'd like to wrap up in the next 13 minutes. To this point, we talked about many ways to change plants genetically. We don't know what changes are made in DNA in any of these cases. We can have massive rearrangements of chromosomes, genes, doubling of whole sets of chromosomes. We don't know how many genes are affected. We just know that we like the outcome. There's maybe requiring special tools and techniques. Solutions can take years, which is important to note, and that dangerous products have been made through these processes. We've created potatoes through traditional breeding that have contained too much of solanine, a compound that can be toxic. We've created other problems too with celery that was bred for insect resistance, that the naturally occurring insecticides in celery would cause human interactions or human allergies. So this is what we know to this point. We also know that all methods can be used in organic production, no problem, and that there's no concern, no testing, no labeling. So from all of the ways in which we've been classically genetically improving plants, it's a very random and wide open process that people are very comfortable with. Now let's contrast that against genetic engineering. First, we'll talk briefly about how we genetically engineer a plant. And it goes back to this idea of, like I briefly mentioned before, about how we can take a cutting from a plant and place it into a hospitable environment. And you'll see the cells on that stem or on a leaf begin to form roots. Oops. And what you're seeing here are these cells kind of reprogramming themselves to understand that their new position in the plant and take on a new role, such as an adventitious root that now can bring water and nutrients into that plant body. This happens in plants because plants are stuck in one place. And in animal development, most changes occur because of time. In plants, it happens because of the environment, where you're placed, the challenges you're confronted with. These changes in cells are something that we call totipotency. This is maybe a little bit hard to see. Basically, the classical model was that you could take phloem cells from carrots, so carrot vascular tissue, and break it into pieces, and then take those little single cells, grow them in culture, turn them into little embryos, and grow a whole new plant from a single cell. Really neat trick. And for years, scientists worked on how to do this. How do you perfect and master this ability to go from a single cell to a whole plant? Genetic engineering is the process of being able to put the gene you want into that single. Oh, here, this is just an example of taking a piece of a plant with some plant tissue and being able to get it to turn into an entire plant or a whole set of plants based upon regeneration from a single cell. So regeneration is this process of these single cells changing their identity and changing into whole small plants or small organs. That's the process that's used in genetic engineering, because if you can place your favorite gene into that first cell, that foundational cell, you can run it through that tissue culture process, create embryos or create organs that then turn into an entire new plant that all contain the gene you installed way back in the beginning. So this is the fundamental idea. Being able to place a gene that you understand into that single cell and then regenerate that entire single cell into a complete plant body. So how do you get that gene into that first plant? Into that first cell? How do you get it into that first cell? And if you look on the internet, you can find a variety of different solutions that they propose. In a big room with lots of people, that usually gets a laugh. But this is what we find online, that there's this kind of yuck factor that's brought into it, that this is somehow syringes and funny colored fluids put on with scary scientists in masks. But really, it's nothing like that at all. There's a couple ways to do that. The first way is to use Mother Nature's genetic engineer, something called Agrobacterium tumefaciens. Agrobacterium has a normal life cycle where it colonizes inside the plant. And part of that cycle is to push a piece of DNA into the plant's DNA. It takes a piece of DNA from it, moves it into the plant cell. And then that plant cell forms these structures that then provide a hospitable home for Agrobacterium. The process is really well understood. And that molecular mess there on the right is just an idea about the way that scientists have taken apart this pathway to understand how this bacterium moves a piece of DNA into the plant cell. Today, scientists have been able to rewire Agrobacterium or change Agrobacterium to be able to, instead of delivering its payload, to push the gene that we want installed into the plant. So in this case, Agrobacterium would be used potentially to infect that first plant cell that we could then grow into an entire new plant containing the new genetic instructions. The other way that this has been done is with something called biolistics, where small particles are coated with DNA and then literally shot in the plant tissue using compressed air or helium, using a charge of helium to propel those particles into plant tissue. And then some of these will get into the nucleus or pass through a cell, dropping their DNA, which is then taken into the nucleus and integrated. This process is much less likely, not used very often. But the idea is to be able to introduce new DNA into the nucleus of that new plant. So we get that, you either use Agrobacterium or biolistics to introduce a new piece of DNA into a single cell. And here's what that process looks like altogether, that once it's in that single cell at the end of the blue arrows, you then will start to see small structures that will occur, and then those will turn into just very early leaves, then maybe whole shoots, and then eventually an entire plant containing your gene of interest. It's not really such a scary black box process. You could come to my laboratory and do this. I mean, we do this every day. Very simple way of introducing new genes into plant tissue. So what do we add? Why do we do it? How do we affect the way a gene behaves by putting it into a background of a plant? And what we do is we either turn off a gene that's already on by introducing something, or we can introduce something to turn on a gene that normally is off or maybe not even there. So here's some basic ideas about how those processes work. The first example is how we can turn off a gene. So this is the idea of interrupting that central dogma of molecular biology. What we're doing is interrupting the process by throwing out the thumb drive. We're essentially breaking the information from going from the hard drive to the printer by throwing away the USB drive. And that's what RNA silencing does. You have the DNA in the nucleus that produces RNA, yet scientists introduce an RNA species that is kind of a mirror image of the RNA that's produced. In that way, think of it as canceling it out. It essentially cancels out that intermediate message. Then you don't produce the protein. You don't have that protein available to underlie the trait of interest. So this idea of gene silencing is very important in the process of saving the papaya, for instance, from viruses. And we'll talk about that next week. The other thing we do is something called overexpression. How do you turn a gene on or maybe turn it on all the time when it normally isn't on all the time? Now, way to think about how this works is genes are turned on and off by using a regulatory sequence that's typically upstream of the gene called a promoter. And in these pictures, it shows two different promoters, one called NAC2 promoter and one called CAMV35S, which is cauliflower mosaic virus 35S. These were placed upstream of something called GFP, which is green fluorescent protein. It's a gene that comes from the jellyfish that when you shine a blue light on it, glows green. What you're seeing in the bottom panel is an experiment that was done to test the activity of the NAC2 promoter compared to positive and negative controls. On the far left, you have negative controls, where either in the presence or absence of ethylene, this organism is not producing the GFP. If you look down the stream to the other positive control, you see 35S, which is turning on GFP in both cases, independent of whether there's ethylene there or not. Ethylene is a plant hormone that changes gene expression. In this case, it doesn't care about ethylene. We like to say that cauliflower mosaic virus promoter is constitutive. It's on all the time. It's on at a high level and in all tissues. Why this is important is because many of today's genetically engineered traits are engineered downstream of a cauliflower mosaic virus promoter, which allows them to be turned on in all tissues at all times. The one in the middle, well, you can see this one is ethylene inducible. That you only see the presence of GFP expressed after it's been given ethylene. No ethylene, no GFP. And that's an example of a regulated promoter. We can mix and match regulated promoters with different proteins and different other sequences, almost like Legos. You pop in one, take out another. You can mix and match through this process of recombinant DNA technology. In most plants, we're overexpressing a gene. In today's, at least in major agronomic crops, we're using cauliflower mosaic virus 35S promoter to either turn on a gene that confers resistance to insects, or maybe it confers resistance to an herbicide. We'll talk more about those next week. The other way to think about genetically engineered plants is to talk about the way in which where the parts come from. And typically when we talk about genetically engineered plants, we'll refer to them as transgenic. And this means that there's been a gene that's moved from another species, or maybe the regulatory control region from another species. In this case, having the cauliflower mosaic virus promoter pushing GFP or driving GFP expression would be an example of a transgenic mechanism. Cisgenic is when we talk about plants with all the parts, all the genes coming from sexually compatible species. And what this means is that if you take an apple gene for resistance to a disease and place it downstream of an apple promoter and move that into apple, that's an example of a cisgenic trait. You are using all apple hardware to change a trait in apple. The really good story from this comes from the disease called apple scab. Years ago, scientists figured out, plant breeders figured out that you could cure apple scab by making crosses of Malus domestica, the modern apple, against something called Malus floribunda, floribunda, which is an apple that does not get apple scab. And you could cross them. And it took many, many years to be able to get a commercial apple that wouldn't get resistance to apple scab, probably 50 years plus. They were able to do it with cisgenic approaches in 5 years, moving the same gene under its own control into a sensitive apple. So cisgenic approaches allow accelerated breeding. The last thing we'll touch on today is this idea of gene editing, and this is something you should become very familiar with. You'll sometimes hear it referred to as CRISPR or also CRISPR-Cas9. What this is these are ways in which you can make precise changes in the sequence of DNA, but leave no hardware behind. So in transgenic and cisgenic approaches, you leave other bits of agrobacterium sequence around. And that's what people tend to find objectionable. In gene editing, it's possible to do it in certain ways that you leave nothing behind except for the change you want. And that's a really important point of that particular technique. We don't know much about how it'll be regulated yet. We'll talk about that later. But in the second part of the lecture, we talked about some approaches where you know the changes in DNA because you were the one who told agrobacterium what to install. They were minor alterations, typically a single insertion in a single part of the genome. We know precisely how many genes are affected, and again, it requires specialized tools and techniques. And we know that the solutions can happen in months to years as opposed to many, many years, such as in other breeding techniques. And thus far, there's no evidence of negative health effects, probably because we understand what genes we're installing and how they're installed. At the same time, these products are not allowed in organic production, unfortunately, and that the products are extensively tested, yet there's fear about them and they're tightly regulated. The last thing I'll show you today is this table that I made years ago that if you look on Google under Frankenfood paradox is a comparison of all these different techniques. And it talks about these traits a little more extensively. What are some examples in the different ways that we genetically improve plants? So really the idea of today's capsule was to give you a sense of the ways that humans have altered plants through time. We moved them from where they belong to places where they don't belong, and we changed the way they look so that they would have better performance for humans. We've been changing plant genetics through domestication. And that's why I really hate the term genetically modified organism. If there's one thing you take home from today, it's the idea that we've been genetically modifying plants in all kinds of ways that we can't predict for a long time. And really only in the last 30 years have we been able to engineer and change genetics in ways that are extremely precise and that we understand exactly what we're changing. Traditional techniques, we can do some pretty strange things. We can use radiation, we can use chemicals, we can alter DNA with damage or by combining things that could never combine naturally. Nobody really seems to mind that too much. Yet, when we use genetic engineering, when we either can have addition of a new gene or suppression of a single gene, or maybe a couple genes, people tend to get a little bit upset. And that's the technology that people are most curious and concerned about. This whole setup today was really important because we needed to understand the way that we change genetics of plants in order to be able to step forward and understand the precise ways that we have changed them and the new traits that are coming. So, I also do a podcast that's worth a listen called Talking Biotech Podcast. It's available at talkingbiotechpodcast.com and also through your places where you can download podcasts. We talk extensively about the domestication of different plant species and plants that you may know, and they all have very interesting stories. So I'll stop there for today. Are there any questions?
Is rice, like the yellow rice, genetically modified?
Yes, all right. So the question was, is the yellow rice genetically modified? So the answer is no. So what you're thinking about might be the golden rice. And golden rice is a genetically engineered rice which has higher levels of beta carotene. It's the stuff in carrots. We'll talk about that one next week. There are other strains of yellow rice. Most rice is yellow because of the addition of colorants. So, but we'll talk about genetically engineered rice next week. Any other questions or thoughts? Sorry, I kind of got on a roll there because I just kind of, it's hard functioning without a big audience, but we will get through it. Thank you for coming. I appreciate it a lot. And anyone who's watching next week, come on down. Let's do it live. Thank you very much. We'll see you next week.